Gary Johnson. I am a professor here at Texas A&M University. And today we're going to talk about muscle, that is the gross skeletal muscle, or the fibers inside the cells, as well as the bands, connective tissue layers, ultrastructure, nerves, and receptors. Another type of muscle is skeletal muscle, and here we see a muscle. This is an arm, a muscle. We can see a fascicle muscle, which is groups of cells. And then we have individual muscle fiber or muscle cells. So this individual cell, you can see it the same over here with nuclei on the periphery, nuclei located here, a big cell. And if you look inside there, you have myofibrils. Myofibrils are linear structures that are inside the muscle fiber. Muscle fiber here with a plasma membrane, nuclei on the periphery, and then we have inside the cell myofibrils. If you look at the myofibril, you can see some dark areas and some light areas. And you can even see a darker structure there, which is a Z disc here and here. And the sarcomere is from one Z disc to another Z disc. And you can see that this is myosin, and the actin, the smaller filaments, is actin. And so it's the location of these in relation to one another that makes the sarcomere contract, as we'll see here in a minute. So if we look at skeletal muscle, we can see the nuclei in the periphery, and we see some capillaries in between there. And here we see connective tissue, and this is the paramecium. The connective tissue that we see here is the paramecium here, skeletal muscle cells, capillaries in between. This is nerve that we see. Nerve is running through here, connective tissue of the paramecium. Now, individual cells have connective tissue in there. We call it endomecium. In a minute, we'll look at that. But remember, what we see here is a muscle cells are innervated, so they can be more than one cell innervated by the same nerve, and so that's what we see here in the muscle. These are the motor end plates where the nerve is coming down, innervating these muscle cells that we see there. One nerve innervates more than one muscle cell. So if we get back to our light and dark areas, we see the dark band A band, and the A band is anisotrophic. In other words, it does alter polarized light. The A band, that's where it goes A for anisotrophic. Isotropic I band does not alter polarized light. And so it's the lighter band in between. And in between there you have the Z disc. So from one Z disc to another Z disc is one sarcomere. And you can see that in contraction, what you have is Z disc come closer together. For relaxation or stretching, they're further apart. And so the sarcomeres are further apart in the case of stretch. In the case of contracted, they're close together. And so if they totally overlap the actin overlaps the myosin, you have it contracted. If there's spaces that you have either actin alone or myosin alone, which is the H band, that is the relaxed state. So we see the dark is the A band and the I band is the light band. So if we look at muscle, here is a piece of muscle. On the outside of the muscle, we see the epimecium, epi outside, way outside connective tissue. But we see divisions inside there, and that's the paramecium. The paramecium that goes in through there. And then if you have around individual muscle cells would be the endomecium. So endomecium is around individual muscle cells that are inside there. So the paramecium is coarse connective tissue. The epimecium is coarse, less coarse with the paramecium, but the endomecium is delicate connective tissue around individual cells, and this is where we saw the capillaries in the endomecium. So this is a fascicle of muscle cells here and here, endomecium in between. So these are nuclei of muscle cells, connective tissue of the paramecium that we see, and indeed the nuclei on the periphery, and it's striated with light and dark bands, the A band being the dark band. If we look here in between these muscle cells and the endomysium, we can see capillaries. These are endothelial cells of capillaries. You can see the capillaries branching as they go through there. You have to be a little crazy like this, I guess, but these are individual muscle cells, and you can see in the endomysium, you can see capillaries that are feeding these cells. If you look at another view of this, you can see the striations very nicely, nuclei on the periphery as you see there, cross-section and longitudinal section, and you can see the A and I band. And at a higher mag, you can see not only do you have the A band, the dark band, the light band is the I band, but you see the Z disc as well. So you can see the Z disc right in through there. From one Z disc to another Z disc is one sarcomere, and you have repeating units of that along the muscle that contracts. One muscle cell is attached to another muscle cell by the endomysium, is what attaches one to another. And so we see electron microscopic view of skeletal muscle cell. Here's a nucleus on the periphery, and you can see the sarcomere from Z line to Z, from Z disc to Z disc or Z line. One sarcomere. The H band is a lighter band where you do not have overlap between actin and myosin. The A band, the dark part here of the A band stays the same. It's the I band that moves. And so here we can see in the esophagus, you have a combination of skeletal muscle and smooth muscle. You can see the smaller smooth muscle cells, the nuclei and periphery of these skeletal muscle cells, nerve, connective tissue in through there. We can see there this is skeletal muscle and smooth muscle. If you start in the beginning, at the top of your esophagus, you have mostly skeletal muscle, but as you go toward the stomach, you have more smooth muscle, so that it changes where you're located in the esophagus. This is muscle that's highly contracted, so you can see the Z disc here, and you can hardly see an I band at all. So when it's highly contracted, the I band almost disappears, as you can see there. And certainly the H band in the middle has disappeared. So you can see the hypercontracted of this muscle compared to the other one that we saw. So I mentioned the motor end plates where one nerve innervates more than one. Also, there's a muscle spindle, which are receptors, a stretch receptor, and it has a connective tissue capsule, and then some of the fibers themselves are located. So these are muscle cell fibers that are inside the capsule. So the motor end plates we saw before, innervating the skeletal muscle cells, but this is a muscle spindle, and you can see this is a longitudinal view of it. This is a cross section of one, but you can see these fibers. If you see a higher mag of these, you can see there's actually striations of these intrafusal fibers that are inside the capsule. The capsule is dense, irregular connective tissue that goes around it. If we look at the tongue, we can see another one. Here is a muscle spindle here, and there's another muscle spindle here, and these are the interfusal fibers right here, and this is the end of one of those fibers, but that's what we see. And this is a stretch receptor in muscle. We want to acknowledge the original source of images, micrographs, drawings that have appeared in this presentation. And we have identified the specific source where the slide was, that image was used. We want to thank the original sources. So this is the end of muscle, that is the skeletal muscle cell, the fibers inside the cell, the connective tissue layers around them, the ultrastructure, nerves, and receptors.